It is a great pleasure to introduce to you Dr. Jiang, a colleague of mine for several years. He did her undergraduate work in China. Post her undergraduate work, she had some very interesting and good training in Japan, where he visited the Japanese equivalent of the National Institute of Health. And then I was lucky, we were lucky, to recruit her to Stony Brook as a graduate student. Beautiful work as a graduate student, and today she's presenting part of that, but also a very important work post her PhD years. She did that alone, but also with the help of very good people in the department in my research. My pleasure to ask you to come.
Thank you, Eka, for the introduction. Ladies and gentlemen, thanks for your coming. It's really a great honor for me to share my research with you today. I hope you will take something with you after my presentation when you leave this room. The title of my presentation is One Stone, Three Birds. Studies of genetic recombination converge enterovirus evolution with global polio eradication and the mechanism of viral morphogenesis. Based on this title, you may expect there are 4 parts in my presentations. One part, the stone, is our studies on genetic recombination of enteroviruses. Based on these studies, we made 3 interesting observations that have important implications to enterovirus evolution, global polio eradication, and the molecular mechanism of enterovirus morphogenesis. Now I will talk the first part, the stone of my presentation. It's our studies on genetic recombination of enterovirus. Enterovirus is a genus of RNA virus that belongs to small RNA virus family, Picornaviridae. Picornaviruses are divided into a number of different genera that includes a bewildering array of human and animal pathogens. Among them, poliovirus, human rhinovirus, foot and mouth disease virus, and hepatitis A virus are probably the most famous. Poliovirus, as a prototype, are best studied and are most famous human enteroviruses. Today, my talk will be focused on human enteroviruses. Human enteroviruses are ubiquitous. Every year, they account for an estimated more than 1 billion infections in the world and 10 to 15 million symptomatic infections in the United States alone. Enterovirus genome is a linear positive sense single-stranded RNA that enclosed in a protein shell called a capsid. The 7.5 kb genome is divided into 3 parts: the 5' non-translated region, the open reading frame, and 3' non-translated region with poly A tail. The 5' NTR and the 3' NTR contain important RNA structures for viral translation and viral genome replication. The open reading frame, which is initially translated into a long single polyprotein, which was further processed by viral encoded proteins into mature viral proteins. The open reading frame can be divided into 2 regions. One is the P1 structure region, the other is non-structure regions. The P1 structure region encodes the viral capsid protein, while the P2, P3 non-structure region encodes enzyme and proteins for RNA and viral replication. For enteroviruses, people think P1 structure region is a primary determinant of the virus identity because the P1 encoded capsid, by binding to specific cellular receptors, determines the tissue tropism and the pathogenicity of the virus. One of the most distinct features of human enterovirus genome is rampant genetic variation and rapid evolution, contributed by both point mutation and genetic recombination. Previous study on virus genome found that during the evolution, point mutation occurs predominantly in structural region, while genetic recombination mostly occur in non-structural regions. There are about 70 serotypes of human enteroviruses. Based on phylogenetic analysis, they are classified into A, B, C, D 4 clusters. Recent years, phylogenetic analysis are extensively used in different organisms to study the evolutionary relationship. To study the genetic recombination, I have been focused on C-cluster human enteroviruses, which includes 11 serotypes of C-cluster Coxsackie A viruses called CCAF, and 3 serotypes of Polio Virus. The grouping of CCAF and Polio Virus in the same cluster indicate that they have very close evolutionary relationship. Poliovirus and the CCAFs are mainly different in their use of cellular receptors that leading to their different pathogenicity. Poliovirus use CD155 as a receptor. After primary infection in gastrointestinal tract, can spread to the secondary target organ, the spinal cord, and cause neurological disease poliomyelitis. CCAFs, by using ICOM1 as a cellular receptor, are commonly associated with respiratory disease like a common cold. Only in rare cases, they cause infantile diarrhea and hemorrhagic conjunctivitis. The reason we choose C-class enteroviruses to study the recombination is because we are concerned about poliovirus. Poliovirus has been one of the most fearful human pathogens in the world, and it has caused and still causing an irreversible, fearful neurological disease, poliomyelitis. To study the recombination between polio and the CCAF, our first aim is to decipher the evolutionary relationship between polio and the CCAFs so that we can predict the emergence of poliovirus and prepare for the prevention and the control of poliomyelitis. Why poliomyelitis brought so much concern? Poliomyelitis, the term was originally come from Greek. It means the infection of poliovirus in the central nervous system can cause the damage in the gray matters of motor neuron, leads to muscle weakness and paralysis. If in this picture, the muscle paralysis happens in the leg of the girl, her leg was so atrophied that he needed the aid of the leg brace to stand and the crutches to walk. The muscle paralysis can also happen in the lung during the acute infection. This picture shows the air-lung field hospitals during polio epidemics in 40s and 50s because the patient need the air-lung to artificially maintain their breathing. The earliest documentary evidence for poliovirus was found from Egyptian stele dating back to 1350 BC. It shows a young man with a atrophied leg and a horse foot, hallmarks of poliomyelitis. At the end of the 19th century, devastating epidemics of poliomyelitis started to occur in industrial countries. And in 1909, poliovirus was identified by 2 Austrian physicians. In 1930s, people found that there are 3 serotypes of polioviruses. And in 1950 and 1960s, 2 kinds of effective polio vaccine were developed. The first one is IPV, the inactivated polio vaccine by Jonas Salk. Later was OPV, the live attenuated oral polio vaccine by Albert Sabin. Since the introduction of these 2 effective vaccines, the burden of poliomyelitis was greatly reduced in the world. And in 1977, smallpox became the first infectious disease that was eradicated. This provided a major momentum to WHO to globally control the vaccine-preventable disease like poliomyelitis. So in 1988, inspired by global smallpox eradication, WHO launched the global polio eradication campaign, mainly by the massive vaccination of the OPV. Since 1988, great progress has been made towards the global polio eradication. From this map, we can see that the number of countries where wild-type virus is endemic greatly declined. And also we can find that the number of wild-type polio cases in red has decreased from 350,000 in 1988 to 1,350 cases in 2010. This great success was mainly achieved by massive OPV vaccination. However, in the end game of global polio eradication, OPV became a double-edged sword. The reason that OPV was initially chosen for the global eradication because compared with IPV, OPV has key advantages. As an oral vaccine without needle and syringe, it's less expensive and easy to administer it. And it's also the OPV also provides better protection to individuals because OPV can stimulate mucosal immunity that is not seen in the IPV, and this mucosal immunity can curtail the spread of the wild-type virus in the individual who take the vaccine. However, as a live vaccine, OPV displays the risk profiles. It is genetically not stable. It has a tendency to revert to the neurovirulent form, so we call vaccine-derived polio virus. They are highly involved in causing vaccine-associated paralytic poliomyelitis. VTPV has been isolated from immunodeficient individuals called IVDPV. A more serious scenario for OPV is its ability to evolve into circulating vaccine-derived poliovirus. Their neurovirulent phenotype and ability to transmit are identical to wild type. That's why they can transmit in a population that is poorly immunized and cause poliomyelitis outbreaks. The first CVDPV was reported in 2002 when WHO studied the poliovirus involved in the outbreaks in Hispaniola. They were startled to find that it is OPV, not wild type, caused the epidemics of poliomyelitis. And more surprisingly, they found that these circulating vaccine-derived poliovirus are recombinants between mutated SABIN1 OPV strain and enteroviruses. Because from the sequence analysis, they found that all these CVDPVs has a genome of similar features. The 5' half of the genome, which contain the capsid coding sequence, are from mutated SABIN strains, while the 3' half of the genome containing the non-structured protein coding sequence are from other non-polio enteroviruses. And the crossover of the recombination occurs in the P2 domain of the genome. And due to the complexity of the patterns in the non-structured region, the identity of the enterovirus involved in the outbreaks cannot be determined. And at that time, no experimental evidence to show the recombination of this type has been presented. Since 2002, CVDP had been reported every year in different parts of the world. As we can see from this map, the 3 serotypes of OPV has all involved in the outbreaks of poliomyelitis in recent years. For some reason, type 2 OPV make more problems. So the emergence of CVD PVs really pose a great challenge to the global eradication, especially at the final stage. So the continuous evolution of CVD PVs reinforced the significance of studying the recombination between poliovirus and other enteroviruses. Therefore, the second aim of our study for genetic recombination between polio and the CCAF is to provide direct experimental proof that CCAF can be recombination partners to poliovirus during evolution of CVD PVs. Poliovirus genetic recombination was first described in 1962 by Hurst. He proposed that polio recombination occurs during genome replication, but at that time, nobody believed him. After 20 years, the experimental evidence for recombination of polio was provided by Agar and Kukerkart. They also calculated the frequency of recombination between poliovirus and found that the frequency is 10 to the minus 6 between the poliovirus of the same serotype. And the frequency goes down to 10 to the minus 6 if the recombination occurs between poliovirus of different serotypes. And it's proved that this difference is due to the sequence homology of the virus, of the parental viruses. In 2007, I provide the first experimental evidence that poliovirus can recombine with non-polio enteroviruses. I will talk about the experiment in more detail in my talk later. How genetic recombination occurs? Kukerkart in 1980s proposed a template switching model to explain the mechanism of a genetic recombination. The first step is the co-replication of a parental virus in the same cell. One parental is called acceptor, the other is called donor. When RNA polymerase of the donor is synthesizing its negative strand, in high homology region where the sequence of the parental virus are very similar, the minus strand of the donor may hybridize with plus strand of the acceptor. This hybridization may make the RNA polymerase of donor so close to the acceptor plus strand that this polymerase may switch its template from donor to acceptor. After the template switching, the donor RNA polymerase may continue its copying activities by using the acceptor strand as a template and generate the negative strand chimeric RNA, half the genome from acceptor, half the genome from the donor. Using this as a template, it can generate the positive strand chimeric RNA, which may finally produce recombinant virus. Based on this model, when the poliovirus and the CAF co-replicate in the same cell, we can expect two possible scenarios. The RNA polymerase may switch from CAF to polio or from polio to CAF. Consequently, it may generate two types of recombinant. Recombinant either with a chimeric genome containing P1 region from polio, but the P2, P3 region from CAF, or recombinant with a chimeric genome that P1 region from CAF, but the P2, P3 region from polio. To test these two kinds of recombinant, we set up an experimental system. For poliovirus, we choose a type 1 Mahoney string, and for CAF, we use CAF 2020 because the moderate genetic distance between these two viruses. We use both the in vitro and in vivo strategy to study the recombination. In vitro recombination can be considered as forced recombination. It's by genetic engineering, the P1 coding sequence of CAF and the polio are swapped so that we can make the chimeric cDNA, PCC, and CPP. In vivo recombination study is the selection of natural recombinant when the parental virus co-replicate in the same cell. In case of our studies, we co-infect the PVM and CAF20 on HeLa cells, which express both CD155 and ACOM1. During the replication, the recombination may occur and generate the progeny with chimeric genome PCC, CPP, besides the parental viruses. So interestingly, from our in vitro study, we observed a parent-specific asymmetry of genetic compatibility. What does this mean? It means if we look at we compare the growth phenotype of PCC with the wild-type poliovirus, as we can find in replication phenotype and the virus yield, the PCC are very comparable to the wild-type polio, indicating that the P1 region of polio is very compatible with the P2, P3 from CAF. However, if we compare the chimeric virus CPP with CAF, we found that CPP show a very severe phenotype. It cannot generate the virus until after 4 passages. And if we look at the virus titer, we can also find that there is 1,000-fold difference. The severe phenotype of CPP indicate that the P1 region of CAF is poorly compatible with the P2, P3 region of polio. That's why we think there is a parent-specific asymmetry of genetic compatibility. To see whether this phenomena is also extended to, is a general phenomena, we extend this in vitro study to 2 other pairs of polio and CAF, which are of either smaller genetic difference or bigger difference than the one I talked just now. One is the one with the smaller distance also shows a little bit compatibility asymmetry. But if you look at the one that has the larger genetic distance, it shows more apparent asymmetry. So this confirms that this phenomena is not specific to PVM and CAF20. It's a general phenomena for polio and CAF. To further determine whether this phenomena we can also observe in vivo, we make 2 crosses between PVM and CAF20. The one cross is between the wild-type polio with guanine-resistant CAF20, so that we can select the recombinant that's the P1 from polio, but the P2, P3 region with the genetic marker is from CAF. The second cross was made between wild-type 20 with the poliovirus with a guanine-resistant marker, so that we can choose the recombinant, select the recombinant CPP, which has a P1 region from CAF, but the P2, P3 region from polio with a genetic marker. Interestingly, the PCC recombinant are easily selected from the cross 1, but the cross 2 never produced a viable CPP recombinant that can be detected. So this again confirmed that the parent-specific asymmetry is a general phenomena between poliovirus and the CCAFs. Because PCC was so easily selected from cross 1, we calculate the recombination frequency between PVM and CAF20, and we found the value is about 10 to the 6th. We think this value is high because it falls into the range of the recombination frequency between different serotypes of poliovirus. By sequencing analysis, we look at the genomic organization of the PCC recombinant, and we found that the crossover point can occur in 2A, 2B, and 2C, which in the P2 domain of the genome. And this is reminiscent of the circulating vaccine-derived polio virus that I showed before. All the crossover regions are also mapped in the P2 genomic regions. The 5' half of the genome is from polio, the 3' half of the genome is from calf. So far, we find that the PCC recombinant are very similar to CVD-PV genetically. So the next question is whether they are phenotypically also similar to CDV-PV. So first we compare it to wild type. And we found that the recombinants with the crossover as 2A, 2P, 2C all grows as well as wild type. And their virus titer are also comparable to the wild type. Then we subject these mutants to the neurovirulence test by using the CD155 transgenic mice. And what we found is their PLD50 value, which is a virus dose that make half of the mice paralyzed or dead, are also comparable to wild type. I mentioned at the beginning that the CVD-PV are very identical to wild type in the neurovirulence phenotype. So based on this, we make the conclusion that the PCC isolate in vivo are also similar to CVD-PV in replication and the neurovirulent phenotype. So based on all this data, I want to summarize my first part. Based on my genetic recombination study, we make the 3 interesting observations. First observation is the asymmetric genetic recombination means the genetically engineered or in vivo selected PCC recombinant consistently survive much better than their CPP counterparts. What does this mean? We ask the question, we think that this asymmetric compatibility may correlate with evolutionary directions within this group. The second interesting observation is that the PCC recombinant, especially the one we isolated in vivo, are genetically and phenotypically similar to CVD-PVs that isolate in the field. So we think this observation is very important because this underscores it reinforces the risk factors associated with the OPV that are currently used. So we think this also have important implications for global eradication because this may drive us to develop a new OPV vaccine. The third observation is that we found that all the CPP chimera yield severe phenotype like C20PP or lethal phenotype like C21PP. So we ask the question, where the genetic block in these chimeras? And this actually leads to very important discovery in the molecular mechanism of viral morphogenesis. So now I will talk about the second part, the first bird, is the implication of our studies to enterovirus evolution. Because we observed the asymmetric genetic compatibility, when you think about the compatibility and evolution, the first thing comes to your mind is probably the computer software. We already know that computer software evolute. The files saved in the old version can be opened in the new version, but not vice versa, because they are not compatible to each other equally. So based on this analog, we also ask the same question. Does the asymmetric genetic compatibility correlates with the evolutionary relationship between these two viruses? And if the answer is yes, we want to ask, what is the evolution direction? Is from calf to polio or from polio to calf? So first, we made the phylogenetic analysis. Because as I told you before, the phylogenetic analysis was recently extensively used to study the evolutionary relationship. And so we first make the phylogenetic analysis in the P1 structure region, which encodes the capsid. And what we found is from the root, usually where we think the evolution starts, the calf and the polio virus, they form very distinct like clusters here. The calf comes first, but seems polio comes later. This indicates that polio virus may evolve from calf by changes in the P1 structure region. If we look at the non-structure region P2 and P3, we found that they are very interleaved with each other in poorly resolved clusters, indicating that the evolution in this region is very constrained. So combine the result from these two trees, it suggests that the poliovirus evolved from a C-calf class ancestor due to the change in the P1 region. Actually, we think our genetic evidence, the observation of asymmetric compatibility, does support this hypothesis. Because if we think about the calf virus, for example, CAF20, as I mentioned that the point mutation is a predominant mechanism that leads to the evolution of the P1 region. So if the point mutation in the CAF20 accumulate to an extent that it may by chance become polio-like, and this virus show a PCC genotype. As we already show in our in vitro genetic study, this virus will grow very well. So that makes the receptor switching from ICOM1 for CAF to CD155 possible. However, if we look at this side, if the point mutation start to accumulate in the polio P1 and the mutation make the virus calf-like, based on our studies, this virus may grow very poorly. That's why we think the chance of receptor switching from CD155 to ICOM1 is not likely. So both our phylogenetic and the genetic evidence supports the notion that the benign closely related C-calves are the ancestor of polio during natural evolution. And the evolution is due to the change in the P1 region that leads to the receptor switching. Now I want to talk about the third part of my presentation. It's the implication of our studies to global eradication. Why we think our studies have important implication to global eradication? Because we already showed that the PCC recombinant are genetically and phenotypically similar to CVD-PVs isolated in the field. And this observation reinforces the risk factors associated with the current OPV. That's why WHO is calling for a novel OPV that is debilitated in its ability to recombine with C-calves. So we have a well-established in vivo recombination system that can be used to design and a characterization for a new OPV. So the question is, how to prevent the recombination between polio and the OPV and the calves? So I already showed that during the recombination, in the high homology region where the sequence of the two parental virus are similar, the R polymerase are easily to switch from one template to the other. That's why the sequence homology, by promoting the template switch, can facilitate the recombination. So we think to prevent if we want to prevent the genetic recombination between polio and the calf, we need to genetically manipulate the region where the recombination easily occurs, and to reduce the sequence homology in this region to the C-calves. So actually the methodology is based on very basic biology concept called the degeneracy of genetic code, also called the redundancy of the genetic code. This means that there are more codons than the amino acid. In other words, one amino acid, like glycine I show here, can be encoded by more than one codons. So so based on this, we can expect that for a protein sequence, we can code it in many different ways by using the synonymous codons of the same amino acid. But we choose a very specific strategy to recode the genome. It's called sequence scrambling. It means we shuffle the synonymous codons within the recoded genome without changing the codon usage, and there is no introduction of the rare codons. So that the codons shuffled in the genome, the amino acid sequence is not changed, but the nucleotide sequence changed. Of course, we cannot do this by our hands, so we need the help from the computer. So we, in collaboration with computer science department on campus, we set up a synthetic biology system in our lab. The blueprint for the recording segments was designed by the computer, and the recorded fragments were de novo synthesized and then cloned into the virus genome. The virus was obtained by transfection of the in vitro transcript of the genome. And I mentioned at the beginning that the P2 region is the hotspot of the recombination. So we focus on the P2 region to reduce the sequence homology in P2 domain. We recoded the P2 region not only to one C cups, we have to recode it against all the C cups. So Dr. Ward from computer science helped me to finish the design by reducing the sequence homology in the P2 to all the C cups. And we found that on average, the homology reduced about 11%. And by plug assay, we found that this virus, even if you have like 493 nucleotide change in the P2 region, still grow as well as wild-type virus. Now we are using our in vivo recombination system testing how much the recombination frequency is reduced by crossing this newly designed virus with all the C cups. We hope that the scrambled sequence here will prevent or reduce the recombination. But we don't know how much is the result yet. So this is a summary from my part 3. Our experiment proved that C-cup can serve as polio recombinant partners, reinforces the risk factors associated with current OPV. The call from WHO for novel OPV drive us to generate an OPV candidate with a scrambled P2 domain will be highly debilitated in its ability to recombine with C-cups, averting the evolution of CVD-PVs. Finally, it's the last part of my talk. The implication of my genetic studies to molecular mechanism of enterovirus morphogenesis. What is enterovirus morphogenesis? It is the final step in the virus replication cycle after viral RNA translation and replication. It involves the encapsulation of the genomic RNA by capsid proteins. Previous study showed that this process is highly specific, because virus have evolved a very neat mechanism to differentiate the cellular RNA from its own RNA. And only actively replicating RNA can be encapsulated. So people ask, what determines the specificity of the encapsulation? There are two possibilities. Initially, people think that there is RNA signals on the newly replicating RNA that can be recognized by the capsid protein and make the engagement of RNA and protein possible. But so far, no RNA signal has ever been found. So people start to think about the protein-protein interactions that may determine the specificity of the morphogenesis. Fortunately, our recombination system just provides a good system to study the morphogenesis. We provide the proof that protein-protein interactions determine the specificity of the encapsulation. Why? Because I already showed that the C20pp chimera display a severe phenotypes. It cannot generate the viable virus only after 4 passages, while wild type can get virus just on the, during the RNA transfection. This strongly indicates that this virus encounters some replication blocks during its replication cycle. So the first block we think may come from the viral RNA translation and polyprotein processing. The second may come from RNA replication, and third may come from the RNA encapsulation. So we look this block one by one, trying to pinpoint where the genetic block is. So it's an in vitro translation system. We compare the polyprotein translation and the processing patterns of the C20pp chimera, and we found they are very similar to the wild type in the processing, indicating this chimera is normal in polyprotein translation and processing. Then we go to see whether the block is coming from replication or encapsulation. So for this, we use reporter virus, which has the Renilla luciferase genes put in front of the, at the N-terminus of the P1 region. After translation, the R-loop protein may be cleaved from the polyprotein and give off the luciferase activities. So using this reporter virus, we can differentiate whether the defect is from replication or from encapsulation. The replication can be evaluated by the transfection of the reporter genomes. As we can see that the chimeric reporter virus are very comparable to the wild-type CAP20 during the transfection, indicating it is very normal in our replication. The encapsulation was evaluated by the luciferase activity when we passage the lysate from transfection to the fresh cells. And we found that the chimeric reporter virus have show no activities, indicating that there is no progeny actually formed during the transfection, even though the replication is good. And this strongly indicates that there is a block after the RNA replication, which should be the encapsulation. So based on this result, we think this chimera is normal in RNA replication, but a defect has defect in encapsulation. I mentioned that this virus, this chimera generated a viable virus after 4 passage. This means the virus has overcome the blocks, probably by getting mutations. So we plug purify the virus from the plug assay and found that there are 2 types of mutants. One type have the mutations in the 2C region. The other is the other type has a mutation in the structure protein VP3. When we put these two mutations separately into the genome, we found that they are partially rescue the virus because we can get the virus on the transfection as wild type, but the titers are still low. However, if we put them at the same time into the genome, generate the double mutant, we found that these two mutations can almost rescue the chimeric virus completely. So this gives us a hint that there might be some interactions between these two proteins so that they can cooperate to overcome the encapsulation defect. So if this is true, so the best cooperating may come from the capsid protein and the 2C protein from the same origin. So based on this, we made another mutant that replaced the P2 of polio here with the CAF20 2C, so that the P1 region and 2C they are from the same origins. And this virus grow almost as well as the wild type, indicating that there is interactions between these two proteins. To confirm this phenomena is not specific to C20pp. We extend this study to a lethal CPP chimera, C21pp. I already showed that in our genetic studies, we found that if we replace the P1 region of polio with CAF21, this virus is totally lethal. Even after 8 passages, we cannot get a viable virus. So based on the previous experiment with C20pp, we also replaced the 2C of this chimera with its own 2C. And we found the change make the rescue the virus. This confirmed the notion that there is an interaction, there might be an interaction between these two, the capsid protein and the 2C. So we use so we confirm the interaction by biochemical analysis. We express the GST2C protein and histag VP3 protein, and we found that the GST2C can easily pull down the VP3. Using as a GST as a control, there is no pull down, indicating that these two proteins does interact with each other. So why we think that this interaction may determine the specificity of the morphogenesis? We proposed a model that of the morphogenesis. It starts with the processing of the P1 capsid protein, which aided by a cellular protein SSP90. The cleaved capsid protein VP0, VP1, VP3 form the 5S protomer, and 5 of the protomers spontaneously form the 14S pentamer. People think that the RNA first interact the pentamer and become nucleation signals that initiate the assembly of the previrion, and the previrion was matured into viral particles by the cleavage of VP0 into VP4 and VP2. So the question is, how the RNA was brought to the vicinity of the pentamers? So based on our result, we think it's in the context of the memory-associated replication complex. It is 2C by interaction with the VP3. Bring the viral RNA to the vicinity of the capsid protein and make the encapsulation possible. So this is the summary of my last part. We found that the genetic block of the CPP is at the stage of morphogenesis, and our genetic evidence strongly support that the involvement of 2C in the encapsulation process, probably by interacting with the VP3. Use biochemical assays, we confirm these interactions. And we propose that these interactions, the protein-protein interaction, determine the specificity of the encapsulation by ensuring that only newly synthesized genome emerging from the replication complex are encapsulated. So finally, I want to conclude make a conclusion for my studies. Based on studies of genetic recombination between polio and CCAF, we provide evidence suggesting a double role of CCAFs in polio evolution. It can serve as a source of ancestor polio virus that originated by point mutations in the P1 region. It also can serve as recombination partners in generation of new polio variants like CVD-PVs. These two scenarios describe the possibility of a continuous evolution of significant pathogen like polio from benign ancestors like CCAF. So we also anticipate that the novel PV with reduced recombination with CCAFs will release the grave burden posed by CVD-PVs to the global eradication. And also, based on genetic and biochemical studies of the CPP chimera, we presented evidence that it is the protein-protein interaction rather than RNA-protein interaction that determine the specificity of the morphogenesis. First, last, is my acknowledgement. First, I want to express my sincerest gratitude to Eckhart. He gave me the chance to work in this lab and to work on this interesting project to help me to grow to be a scientist. Without his patience and insightful guidance, all this would not be possible. I also want to thank the people from Weaver Lab who make the lab as warm as a family. And especially for my project, I want to thank Aniko for her continuous help through these years on the scientific parts and also for my English parts. For the morphogenesis study, I want to thank Ying for her contribution to the genetic evidence, and Chenlin for her contribution to the biochemical evidence. And I want to thank Stefan for his contribution for making this creative reporter virus. Without this reporter virus, the encapsulation study was not possible. Also, I want to thank our previous lab members. Hidemi Toyota was a postdoc in our lab. He did all the neurovirulence tests of my recombinant on the CD155 transgenic mice. And Elizabeth contributed the infectious CDN clone of CAF21. And also, I want to thank the collaborator on campus. Especially, I want to thank Charles for working with me on design of the P2 scrambled virus. And also, I want to thank the collaborator from the Netherlands. They contribute the phylogenetic analysis of the C-cluster human enteroviruses. Finally, I want to thank you, all of you, for coming. I'm taking questions.